Hello everyone, we are from Group 3 with me, Rizky Darmawan, and my friend Raidhan Ahmad Desta, Marian Wisnu, Angelita Rahma, and Farij Maulana are going to do sampling and preservation of some animal species in rice field ecosystem. So let's take a look.
Okay, so first thing first, we are going to some preparation. Today, we bring some equipments like sweep nets to catch the insect, papillate paper to kill the insect, fish net to catch the aquatic animals like fishes or amphibians, and don't forget by the phone to documentation. Alright, so cause we're just examination about the invertebrate and vertebrate animal, so we're using purposive sampling technique today. And to take the sample, we divided into two teams. The one team are going to exploring the right side and the other one exploring the left side. So this technique called plot sampling technique.
And then we are continuing to the next or called preservation time. In this step, we are going to do two types of those, which are dry preservation and wet preservation. But before that, we have to kill some vertebrates and insects with hard thorax in the killing bottle that's filled with chloroform. After that, we inject the animal with chloroform to prevent rotting in animal. And voila! The animal is ready to become a specimen.
The dry specimen technique is capable insect and aragonite. In this step, we mounting the specimen into styrofoam and arranging the body position of animal like when they still alive. First step, we make the base construction for the wings of the insect. Here we using two styrofoam that arranged like trapezium block. After that, we pinch the thorax of insect and put the insect between the two styrofoam. After that, we expose the wings in 90 degrees from the body and cover with papillate paper. So on with the other specimen. For wet specimen, we just put the specimen into collection bottle that filled with alcohol, reduce the water in specimen tissue.
Labeling step is divided into three types: sampling labeling, identification labeling, and display labeling. Sampling labeling is done when sampling occurs. The labels are filled with collector initial, collection sequence, date, and coordinate location. This label is dated up in the feet or abdomen of animals. For the identification label, we put local names, species name, collector initial, date, and place identification. Of course, the label were attached in the styrofoam or collection bottle.
For the identification, we use three steps to identify the animals, which are description, determination, and identification. In the description, we are describing the morphology character in every specimen. After that, in the determination, we are comparing the morphology character that we have with some references like GBIF or journals. After that, finally, we decided the taxon of the animals. And for the result, the high taxon that we identify is order, and continued by family and also species. In invertebrate, the order Ordonata have one species, which are Anisoptera, or in local name Capung. In order Squamata or vertebrate, we have Eutropis multifasciata, or we call Kadal. And for invertebrate, the most species we found is in order Arthropoda with species Phalanga nigricornis, Acrida conica, and Phalanga irregularis, or we call belalang. And for order Lepidoptera, which is also invertebrate, we found Danaus plexivus or called kupu-kupu.